Oke, selamat berjumpa kembali bersama saya. Pada pertemuan ini kita akan membahas materi terkait dengan foraminifera. Sebelumnya, apakah kalian sudah mengetahui foraminifera itu apa? Oke, foraminifera secara terminologi adalah suatu organisme satu sel yang memiliki cangkang kalsit dan merupakan salah satu organisme dari kingdom protista yang sering dikenal dengan rhizopora atau kaki semut. Foraminifera berlangsung hidup secara akuatik, terutama hidup di laut, memiliki satu atau lebih dari bilik yang terpisah satu sama lainnya oleh adanya sekat atau septa yang ditembus oleh banyak lubang halus atau foramen. Cangkang atau kerangka foraminifera merupakan petunjuk dalam pencarian sumber daya minyak, gas alam, dan mineral. Foraminifera juga merupakan sahabat dekat dari amuba, hanya saja amuba tidak memiliki cangkang untuk melindungi protoplasmanya. Kemudian di mana kita bisa menjumpai foraminifera? Menurut habitatnya, foraminifera dibagi menjadi foraminifera bentik dan foraminifera planktonik. Nah, pada umumnya foraminifera bentik itu hidup di dasar perairan dengan substrat pasir. Nah, sedangkan untuk foraminifera planktonik dia hidup pada kolom perairan. Oke, next. Nah, habitat makro foraminifera meliputi seluruh zona dalam samudra. Secara umum, foraminifera dapat ditemukan mulai dari zona neritik hingga batial hingga abisal. Ya, salah satu contoh dari habitat mikroforaminifera adalah permukaan sedimen dan permukaan rangka karang bagi biota epifauna, badan sedimen bagi biota infauna, helai daun atau batang alga dan lamun bagi biota epifauna. Menurut Corliss 1985 menemukan bahwa foraminifera bentik yang hidup di laut dalam ditemukan pada sedimen hingga kedalaman 15 cm ke dalam permukaan dasar laut. Kemudian kita masuk ke tipe-tipe lingkungan habitat makro foraminifera. Yang pertama, foraminifera mempunyai habitat di daerah perairan tawar. Kemudian foraminifera habitatnya di paparan benua juga atau continental shelf atau disebut juga dengan laut terbuka. Kemudian selanjutnya di perairan payau, estuari dan mangrove. Yang keempat, paparan karbonat dan terumbu karang. Dan yang kelima di wilayah laut dalam atau lereng benua hingga zona abisal. Nah, salah satu contoh dari habitat mikro adalah permukaan sedimen dan permukaan karang bagi biota epifauna. Badan sedimen bagi biota infauna, helai daun batang alga, dan lamun bagi biota epifauna. Corliss 85 menemukan bahwa foraminifera bentik hidup di laut dalam dapat ditemukan pada sedimen hingga kedalaman 15 cm. Data sebaran memperlihatkan terdapatnya dua kelompok biota yang memiliki cara hidup berbeda, yakni epifauna dan infauna. Kemudian Corliss 85 menyebutkan bahwa kedua kelompok ini memperlihatkan perbedaan tipe morfologi pada cangkangnya. Nah, apakah foraminifera mempunyai manfaat? Jelas, foraminifera mempunyai manfaat. Yang pertama, foraminifera memberikan data umur relatif batuan sedimen laut. Kemudian manfaat yang kedua, foraminifera memberikan data tentang lingkungan masa lampau. Itu berdasarkan skala waktu geologi. Kemudian yang ketiga, foraminifera dimanfaatkan untuk menemukan minyak bumi, gas alam, dan mineral. Berikut contoh dari foraminifera. Ini merupakan contoh dari foraminifera planktonik. Ya, dengan bentuk yang seperti ini, dia lebih bundar. Oke, next. Kemudian kita masuk ke fosil index. Untuk mendefinisikan batas dalam skala waktu geologi, dalam korelasi strata, ya, fosil indeks akan sangat membantu. Nah, karakteristik fosil indeks yang baik menurut Ghost 2006, yang pertama, dia mempunyai khas dan umum. Kemudian tersebar luas dan mudah dikenali atau didiagnosis. Kemudian yang ketiga, rentang waktu geologis yang terbatas. Yang keempat, jumlahnya berlimpah. Dan yang kelima, mudah diawetkan. Serta yang keenam, ditemukan di beberapa lingkungan. Ini merupakan contoh dari fosil indeks, ya, fosil indeks dari Foraminifera. Next. Sekian dan terima kasih.
Foraminifera are single-celled protists that range in size from that of a grain of sand to a few millimeters long. Foraminifera make shells of calcium carbonate, or build their shells from sedimentary grains in their environment. These shells readily preserve in marine sediments, and we can find these microfossils from 570 million years ago to the present day. This makes them a wonderful tool for reconstructing ancient environments. Because different species of Foraminifera lived at different times over their 540 million year history, identifying the Foraminifera that are preserved in a sediment sample allows micropaleontologists to determine the geological age of the sediments. This process is called biostratigraphy and is important for relating distant bodies of marine rock to each other as we piece together the Earth's geologic history and for tracing economically important petroleum source rocks. Different species of Foraminifera also prefer different environments. Thus, the species we find in a sediment sample gives paleontologists the ability to reconstruct changes in oceanic conditions through time. Foraminifera can be either planktonic or benthic. Planktonic Foraminifera live suspended in the water column and are especially sensitive to changes in ocean temperatures. These Foraminifera are instrumental in recording glacial and interglacial climate changes over hundreds of thousands of years. Benthic Foraminifera live on the seafloor or within the seafloor sediments. The fossils of benthic Foraminifera are very common in the sediments of shallow ocean depths. In addition, different species of benthic Foraminifera live on the continental shelf, the continental slope, and on the abyssal plain. Knowing which species occur at which water depths in the modern sediments allows paleontologists to use the remains of Foraminifera to determine past changes in sea level along coasts. With this data, they can predict how sea levels will change in the future. Different types of Foraminifera also respond to changes in food and oxygen availability at the seafloor. By examining the changes in Foraminifera in ancient sediments, we can reconstruct how oxygen and food availability changed with ancient climate changes and understand how modern climate change will affect ocean ecosystems. This video will focus on the morphological characteristics of benthic Foraminifera that micropaleontologists use to differentiate among different species and reconstruct ancient environments. Among benthic Foraminifera, the first characteristic important for identifying major species groups is the material from which the Foraminifera makes its shell. Agglutinated Foraminifera embed sediment grains and the remains of other organisms into an organic matrix to make their shells. Both hyaline and porcelaneous Foraminifera use carbonate to make their shells, but porcelaneous Foraminifera have a smooth, opaque shell, whereas hyaline Foraminifera are dotted with pores and often translucent. Just identifying these different shell types can help reconstruct the salinity of ancient bays and estuaries. Porcelaneous Foraminifera are most abundant in hypersaline environments, while agglutinated Foraminifera dominate brackish water environments, and hyaline Foraminifera are most common in normal marine conditions. The shapes of the overall Foraminifera are very diverse, and is the second thing you should observe when trying to identify a species. Foraminifera may have a shape like a vase, or a cone, or an arrowhead, or a lens, an oval, a disc, or a fan, among many other potential shapes. As you examine the overall shape, you may need to look at the Foraminifera from a few angles to understand its shape in 3 dimensions. To help you visualize the Foraminifera in 3 dimensions, the images you will see will show 3 views of each specimen. In the form shown here, you see its umbilical side, an edge view with the aperture facing outward, and its spiral side. Most Foraminifera grow by adding chambers to their shell to accommodate the growing cellular material that makes up the organism itself. Sometimes the chambers are shaped like short and broad boxes, and other times they are long and thin, with the long axis of the chamber parallel with the long axis of the overall foraminifer. The chambers may also be inflated and globose, with rounded chamber walls shaped like arches, or have walls that meet at sharp angles. Some foraminifera have only one chamber, while others may have dozens of visible chambers. The first initial chamber, or proloculus, of a foraminifer is nearly spherical in shape, and chambers are added as the foraminifer grows. The pattern by which chambers are added determines the overall shape of foraminiferal shell and varies across genera. This is one of the most important morphological characteristics to recognize when trying to identify a foraminifer. Unilocular foraminifera have only one chamber. Others add chambers in a single row or column and are called uniserial. Biserial foraminifera have two rows of chambers added on either side of a central axis. Triserial foraminifera have three rows of chambers, which sometimes appear to twist around each other as the chambers get larger over time. Some foraminifera coil in two dimensions, creating an expanding spiral outward from the initial chamber. These are called planospiral. If the coiling is offset in a third dimension, coiling outward and upward at the same time, this creates a cone shape and is called a trochospiral chamber arrangement. Some foraminifera even combine these arrangements, transitioning from triserial to biserial over the lifespan of an individual, or adding chambers along two rows like a biserial while also coiling. These different morphologies reflect the different lifestyles of the foraminifera and are related to the environments in which they live. For example, biserial and triserial foraminifera often live deeper within the sediment than other forms and are more tolerant of low oxygen conditions. The aperture is another important characteristic for identifying foraminiferal species. This opening is what allows the cellular material inside to communicate with the outside environment to gather food. It is associated with the youngest chamber of the foraminifer. The shape of the aperture varies among genera from round to an elongate loop to a thin slit. Some genera have apertures made of multiple pores or many slits radiating outward from a central point. The aperture may be located at the end of the youngest chamber, on the face of the youngest chamber, or tucked along the chamber margin. As I'm sure you've noticed, foraminifera have a great deal of morphological variation, especially given they are single-celled organisms. As you learn to notice and identify these different shapes and forms, you may also notice differences in ornamentation that can help you identify different genera and species. Sometimes the aperture will be set on the end of the neck that extends out from the chamber wall. Or if you look down in the aperture, you will see small projections or teeth. Or you may notice ridges and bumps on the chamber walls that help you to recognize the genus or species. Keep an eye out for those details as you explore the morphology of foraminifera and use their morphology to decode ancient environments.
Foraminifera, or forams for short, are single-celled protists. A protist is a eukaryotic organism that is not a plant, animal, or a fungus. Forams are primarily found in marine habitats, and they are very important in marine food webs. Thousands of species have been identified. Some species are planktonic, meaning that they float in the water column, while the remainder live on or in the sand, mud, rocks, and plants at the bottom of the ocean. They are found in all marine environments, from the intertidal zone to the deepest ocean trenches, and from the tropics to the poles. Specific species can be very particular about the environment in which they live. Some are abundant in the deep ocean or on coral reefs. Others only live in brackish estuaries or intertidal salt marshes. The majority of species have shells called tests. The simplest forms are open tubes or hollow spheres. More complex shells are commonly divided into chambers that are added during growth. The shell may be made of many different materials, such as organic compounds, sand grains, and other particles cemented together, or crystalline calcite. The shell may have a smooth or rough surface. Fully grown individuals range in size from about 100 micrometers to almost 8 inches long. One group, the xenophyrophores, can grow to several inches in size as a single cell. A single forum may have one or many nuclei within its cell. Forums eat food such as dissolved organic molecules, bacteria, copepods, and single-celled phytoplankton like diatoms. Some species that live near the surface have a symbiotic relationship with unicellular algae that they farm inside their shells for food. Others that live in low-oxygen environments form symbiotic relationships with chemoautotrophic bacteria. Some species are parasitic. Forums can move and catch their food with a network of pseudopodia that are thread-like extensions of the ectoplasm. Under the microscope, these pseudopodia appear granular. The pseudopodia are called reticulopodia. They extend from one or more holes in the shell. The pseudopodia transfer material to the inner shell and back. They can be extended and retracted. Bottom-dwelling foraminifera may also use their pseudopodia for locomotion and anchoring. Some benthic species slowly burrow through sediments at speeds of up to a half-inch per hour. Others attach themselves to the surface of rocks or marine plants. Bottom-dwelling forums play a significant role in the benthic carbon and nitrogen cycles. Forums have many predators such as worms, crustaceans, gastropods, echinoderms, and fish. Some species only live a few weeks, while other species live many years. Growth can be continuous or episodic. Growth is obtained by increasing the size of a single chamber or by the addition of new chambers. Foraminifera can reproduce both sexually and asexually. The fossil record of forums extends more than 500 million years. Shells of foraminifera have accumulated in layers of sediment below the seafloor of the open ocean and in areas where the ocean once covered the land. The pink sands of some Bermuda beaches get much of their color from the shells of foraminifera. Since different species are found in different environments, paleontologists can use fossils of forums to determine environments in the past. Fossils of forums have been used for things such as mapping past distributions of the tropics, locating ancient shorelines, and tracking global ocean temperature changes during the ice ages. Carbon emissions that contribute to global warming are putting stress on forums. As more carbon dioxide dissolves in the ocean, the water becomes more acidic. This makes it harder for forums to create the shells that they need to survive.